as promised, we're back with proteins. Proteins being one of those macromolecule groups that we talked about at the very start of this chapter. They are the most complex molecule known in existence today. Hundreds to thousands of different kinds of proteins exist that we're aware of. The polymer is the protein or the polypeptide. The monomer is the amino acid. So amino acids are the monomers of proteins. There are only 20 different amino acids that make up all of the proteins. So it's like, think of it like the alphabet. There are 26 letters in the alphabet, right? So think of all of the different words and the combinations of different words that you can make using just those 26 letters. You can do the same thing with proteins. I've got 20 different amino acids, and I can put them together in any sort of combinations to get different proteins. Again, because I have so many different possibilities of arranging and combinations of amino acids, I have so many different structures, I'm going to have a myriad of functions from this class of macromolecule. They have, again, a myriad of functions. I've put the big ones up here on this slide, but proteins are going to account for more than 50% of the dry matter in most cells. And we can have a structural protein like keratin. We can have a storage of amino acids like in albumin, which is a plasma protein, or casein. A transport protein that you're probably all familiar with is hemoglobin, the transport protein in your red blood cells that carries carbon dioxide and oxygen. We can have a signaling protein like insulin, or we can actually have a receptor for stimuli. So even the receptors within the cells that allow us to bind thyroxine are going to be made of protein. Something you're very familiar with is movement and your muscle, your skeletal muscles. The actin and myosin fibrils, those are made up of a protein, and that's what allows your muscles to contract and relax. Your immune system relies in part on proteins and globulins. Globulins are going to be the proteins that form antibodies. And then even enzymes. So we've mentioned enzymes previously, but enzymes are going to be catalysts that help to drive those dehydration synthesis reactions that we've talked about. So just a vast myriad of functions that we'll see from this class of macromolecule. Here's an overview. This is straight out of the textbook. I'm not showing you anything magical here. Just another way to give you that information. Maybe it helps you to see it in this format over the last slide, but just another way to look at that overview of functions of the protein class. Remember that we said that the polymers are polypeptides. They're built from that same set of 20 amino acids. Amino acids are the monomers. We join them together in a linear sequence. And then we actually have some folding and some bending and some conformational changes. So a protein actually consists of one or more polypeptides that gets folded into a specific design or shape, if you will. Whiteboard question, real quick one, hitting this one right off the bat. You want to be able to tell me what the monomers of proteins are. You want to be able to put these guys, these four terms here, in order. Amino acids, polypeptides, molecules, and proteins, from smallest to largest or largest to smallest. Okay, so amino acid monomers. We need to look at amino acids and what comprises an amino acid. Let me get that cursor out of your way. So all amino acids are going to have a centralized carbon. They're going to have an alpha carbon right there in the center. Off of that centralized carbon, we're going to have 4 covalent bonds. So the easiest one to remember is we're going to have a hydrogen come off of that group. The next thing we're going to have is we're going to have a carboxyl group. Remember, a carboxyl group is one of those functional groups that I asked you to take a look at. We're also going to have an amine group. an amino group. That's a nitrogen with 2 hydrogens covalently bonded to it. These 3 things are always going to be consistent across every single one of the 20 amino acids. Remember, we were talking about covalent bonds here. Okay, and then the variable portion of amino acids is going to be this R group, this side chain. That's what's going to differ amongst the 20 different amino acids. So they're all going to have a central carbon. They're all going to be, that central carbon is all going to have a hydrogen, a carboxyl, and an amino group bound to it. And then it's going to have one of 20 different options. And those are going to be the side chains, or the variable, or the R group. There's the prettiness, that is the 20 amino acids. They break out into nonpolar versions, polar versions, those that are electrically charged, being either acidic or basic. No, you do not have to memorize them all. When we use the amino acids later in the semester, and actually I'm not even sure we do in 181. If we needed to use the amino acids later, though, you would be provided with a chart, so you don't have to memorize those. What you do need to know is the things that we talked about on this last slide about what comprises an amino acid. You want to be able to name the 4 components of amino acid, and you want to be able to draw some sort of generic version. You don't have to draw any of these specifically, but if I were drawing a generic version, it would look a lot like this. Okay? Amino acids as polymers. So the polymers of amino acids are polypeptides, and they are linked together by peptide bonds. So we've talked about glycosidic linkages, we've talked about ester linkages, and now we're talking about peptide bonds. Polypeptides are varied in their size and their shape. So polypeptides can range in length from just a few amino acids to more than 1,000 amino acids. And each polypeptide, remember we're talking about 20 amino acids here, so that linear sequence of different amino acids is what's going to give it its unique function and shape. One specific type of protein that we'll talk about, polypeptide that we'll talk about, are enzymes. Enzymes act as catalysts, and the role of a catalyst is to speed up a chemical reaction. So not all catalysts are proteins, but when we talk in biology and we talk about enzymes, we are talking about a specialized protein. There's a lot of really great things about enzymes. One of them is that they're not changed by doing their job. They can function and perform their function repeatedly over and over and over again, hundreds, thousands, tens of thousands of times per second. They're just a functional workhorse that goes from point A to point B, and they speed up a chemical reaction or a process. And that's a beautiful thing about enzymes. We'll talk again more about enzymes in chapter, 7 or 8 when we start talking about energy. Remember that protein structure and function are tied, just like every other macromolecule, and the lipids that we've spoken about. Functional proteins consist of 1 or more polypeptides. They're going to be twisted, folded, coiled into a unique shape. We talk about primary, secondary, tertiary, and quaternary shape to proteins, and what determines that structure, that shape, is the sequence of the amino acids. And that same structure that gets determined by the sequence is what's going to determine the function. So what you've got right here are 2 proteins. Over here on the right, this is the protein from a flu virus. See how it's bent and folded and moves back on itself? And over here on the left, you have, with this blue and this red, you have an antibody protein that is designed and structured so that it can match up and attach to the protein from the flu virus. It's going to bind to it, and what we're going to see is an immune reaction. Again, 4 levels of protein structure. We're going to come back and grab that in just a moment. We're almost at our 10-minute mark, so I'm going to break this video here, and we're going to come back and look at the definition for the different levels of protein structures.